Hey, hi, good morning, everyone. So congratulations on getting through the first exam of the semester. So your TIs are busy grading the exam now, and they'll get them back to you as soon as they're done. Okay, so we're holding off until both parts of the exam are completed in terms of grading to give them back. All right, so you can look forward to that. This week also, I'm going to send out a TI evaluation survey, and it's all anonymous, but this will allow you to provide your TI feedback on how he and she is doing so far in recitation. They're anonymous. Your TI won't know, your name. But once we've collected them all, I will meet one-on-one with the TIs and go over the comments and work with them to help guide how they might sort of improve the recitation experience for all the students. So look out for that. It's very important that you take a couple minutes and provide feedback, because after all, right, it's based on that feedback that myself and your TI will know if any adjustments can be made in how the recitations are led. All right. Okay, good. Let's go ahead and move on to our new set of topics here. So we're going to begin talking about how genes are regulated in our cells. In other words, how our cells decide what genes are actually used in terms of the cell carrying out its functions. So this is what we call gene regulation. And this is a pretty complicated process, but one that is very fundamental to understanding how it is that our living cells can actually make use of the genetic information that's encoded for in the DNA. So I think by now, all of you have heard this kind of, a notion or concept that our DNA is like the master blueprint of us. Right? And in this class so far, we've begun to appreciate that indeed DNA is the master blueprint for all life on Earth. And we've spent quite a bit of time understanding the structure of DNA and the nature, or at least an introduction to the nature of genes. So certainly, I think ever since high school, you're already familiar with this concept, right? That DNA is this blueprint. But what does that really mean, right? A blueprint implies that it contains information that's needed for planning and carrying out certain types of instructions that result in something, being made, right? And of course, that thing that's being made in this class, what we're talking about, is a living cell. So let's use this blueprint kind of analogy and work with it. Right? So, oftentimes blueprints, you think about blueprints as being things for like buildings. So let's imagine that you are seeing that a house is going to be built or a large building is being constructed. And let's think about the set of master blueprints for that. So a set of blueprints that are put together by, let's say, an architectural firm would contain all the information for how to build this house. And then it's up to specific contractors, the building contractors, to look at this blueprint and carry out their specific jobs. In other words, what each of the contractors specialize in. So in the master blueprint for a building, you'd find instructions, of course, for the foundation to be laid, for carpenters to do the woodwork, the framing, etc. Masons to lay brickwork for building walls, then specializations like plumbing, electrical, and, for example, installation of windows. Right? So consider that each of these contractors, they have access to the master blueprint that has everything on every page, because every page shows the layout of how the framing of the house has to be, where you have brickwork, where the plumbing has to run through the walls and in the floor, likewise all the wiring. But these contractors each only focus on the parts of the plans that are relevant to their job. Right? But they have to do it within the context of working and adjusting and reacting to those around them. Right? Because any small adjustment in, let's say, the framing or the layout of the plumbing, right, might have an impact on things like the wiring or how the windows go in, etc. Okay. So if we think about the idea of a blueprint, then the master blueprint of our cells would be our genome. In humans, that consists of our 22 pairs of autosomes and pair of sex chromosomes. And as we've discussed already in this class, each chromosome is just one long strand of DNA. Right? And along that strand of A's, T's, C's, and G's, you'd have certain regions that represent specific instructions. These are sequences that contain information that are meant to be converted at some point into some other molecule that's going to be assembled based upon that sequence of A, T, C's, and G's, right? And so those regions of the chromosomes we refer to as genes. Right? Genes are stretches of DNA that code for some sort of molecule to be made based upon that sequence. So likewise, if you consider that every nucleated cell in your body has a copy of your genome in it, that means every nucleated cell in your body has a copy of the master blueprint that contains all of the instructions that are required to build a human being. And so the important thing to keep in mind here is, just like those different contractors working on building the house, different cells have very different sort of specializations in order for them to do their job. So that means they're all referring to the master blueprint, but they're only following the sets of instructions that are relevant for them to be the type of cell that they need to be. So in this set of photos down here, I think these are some very beautiful images. These are all different examples of human cells that you would find in your body. And so I've mentioned this before, but the human body consists of around 220, 240 discretely different types of cells. Okay? So within each of those cells would be a copy of your genome. And that genome, among the 22 pairs of autosomes and pair of X chromosomes, so our 46 individual chromosomes that are contained within the nucleus, right, are thousands of genes, right? 20 to 25,000 genes, or 20 to 25,000 individual sets of instructions, right, for making something. Remember the cookie book analogy, right, where I said a cookie book is like a set of 100 different recipes that can be thought of as 100 specific sets of instructions to make a different type of cookie. In our genome, what are we talking about? We're talking about maybe 20 to 25,000 specific genes that code for different proteins. And just think that this wonderful diversity of different shapes, sizes, and functions of cells, right, are all dependent on what subset of genes they're actually using out of your master genome. So remember, every single one of your cells that has a nucleus carries a complete copy of your unique genome that was given to you by mom and dad. Right? And so different cell types in your body will use different sets of genes that define how they appear, where they reside, and how they function, right, in the tissues of your body. So this process by which a single cell will selectively decide what subset of those thousands of genes it actually needs to use and make the products from those genes, that's the process that we call gene regulation. Right? And that's what we're beginning to study with our lecture today. All right. I think a wonderful example of how fundamentally important gene regulation is, has to do with how complex multicellular organisms develop from a single fertilized egg. Right? So obviously, you know that 20 plus years ago, right, you started out life as a single fertilized egg, and that one egg has now become 20 plus years later, right, 30 trillion cells, right, that make up you sitting there watching this lecture. And of course, it's no surprise to you when you look at your body, right? And you realize there's so many different types of cells. They all came from that original fertilized egg, right, that combined the two genomes, half from your mom, half from your dad, to give you that full complement of 25,000 genes that are uniquely you. So I think it's interesting and exciting to start from the beginning and just understand how, as one fertilized egg starts dividing, how very early on these important decisions about what sets of genes are being switched on or switched off in different cells will have a profound effect on how these cells divide and change into different tissue types of the body. So let's look at a relatively simpler organism compared to something as complicated as us, a human being. Let's look at a Drosophila, right? A fruit fly. Fruit fly that you probably don't really think of that much. You probably think of flies as just being a nuisance, right? Like when you're trying to have a picnic and you're trying to continuously chase the flies away from your picnic, right? With your branch or leaves or whatever. But let's look at this. So this image on the top here, right, is an early developing embryo of a fly. So at this point, it's made up of hundreds of cells already. And so what we're actually looking at is a computer-generated map of the locations of these several hundred cells that at this point make up this developing Drosophila larva. That's maybe just like a grain of rice in size and shape. Right? So what are all these colors that you see? Hopefully, by now, you realize, right, that the molecules that are carrying out these different decisions about what sorts of events are taking place to make one cell start to be different than another type of cell, right, are proteins functioning as molecular machines, right, that are interacting with other molecules and carrying out different processes. Where do these proteins come from? Well, hopefully by now, since you just finished taking the first exam in the course last week, you know, if you backtrack, a finished protein, right, comes originally from a messenger RNA that was translated by something like a ribosome into making a string of amino acids. That's the finished protein, right? But you backtrack, that mRNA came originally from a primary RNA transcript that was like a photocopy of a gene, right? And then that original gene then is in the fly's genome. Right? So what we're seeing here are researchers have generated different ways of detecting different types of early developmental proteins inside of this Drosophila. What they've done is they've designed different antibodies that can bind to different variations of a family of important developmental genes known as homeobox genes or Hox genes. And so when the fly embryo is only a few hundred cells large like this, if they then proceed to stain or label all of these cells based upon what type of protein is being expressed in them, right? And each one of these antibodies that is very specific to tagging a particular protein carries with it a different fluorescing kind of marker molecule. Right? This is the pattern that you see. And so you can already see that within a few hundred cells, already the cells are beginning to change from each other to have different expression patterns, right, of different homeobox proteins. So different genes are turning on and making specific proteins, and these early proteins, right, are deciding the fate of these different cells. So things like the appearance of where the eye is eventually going to be, where the mouth end is versus the tail end over here. And in between, this is really striking, right? The different segments that make up the fly's body. You can really appreciate that these cells are beginning to change from one another, right? By expressing different sets of genes that are ultimately going to make these proteins that interact with specific other proteins and allow the adult fly, right, to have all of these important body structures, legs, eyes, mandibles, wings, etc. Right? So from just a few hundred cells in this image to a few million cells, right, in the fly. So how does this happen? We now know that there is a set of genes in the fly genome, shown here as these different colored blocks, right, that belong to this homeobox or Hox family, right? And these different genes produce proteins that if a cell produces this protein, it begins to change the cell, sort of encouraging the cell down a particular pathway of becoming a specialized tissue. So in light green here, right, maybe these genes, when they're expressed in a group of cells that were on this end of the fly, then the protein products of these genes, right, begin to function as little machines, and they favor or interact with other genes so that these cells become like the mandibles or the jaw tissues of the cell. Likewise, in the middle, like this purple homeobox gene, right, if it is turned on in one of these early cells that are in the developing embryo, right, the eventual consequences is the homeobox protein it makes starts preferentially talking to different genes, right, eventually leading to the expression of a multitude of other genes that produce other proteins that are specialized for forming the wings, et cetera, right? And the yellow blocks down here are simply a reminder that it's not like the fly invented these genes. By the time in evolution that you even get to something as complicated as a fly, right? These genes were already part of living things on our planet, multicellular living things, right? And it's just the fly in that particular step in the direction of becoming flies in evolution, right? These genes got tweaked a little bit through mutations or whatever, so that eventually the end result is you get a fly. Right? So you say, okay, well, that's cute, but what does that have to do with something like way more complicated than a fly, right? Because I mean, there's a little fly that you swat without even thinking about. What about something like, I don't know, a person? But it turns out, right, this is a wonderful reminder that we are multicellular organisms that evolved on Earth, just like flies are multicellular organisms that evolved on Earth, right? And so these same ancestral genes that gave rise to things like the different parts of a fly, right? Our branch of evolution had these genes as well, because us and the fly, if you go far enough back in time, eventually we have a common ancestor, right? But of course, I'm talking about like hundreds upon hundreds of millions of years ago. But our branch, right, we took these ancestral genes and they got tweaked over time, right? Eons of evolution. And so we now have our group of human homeobox genes, right? The colors, the fact that they use the same colors as the fly, that's not a coincidence. That's not a mistake. It's to remind you that they are essentially the equivalent versions of the fly genes, right? And they give rise, just like how the fly's body plan is dependent upon those genes, right? So are our body plans, right? So in early embryonic fetal development, these same homeobox genes are critical for laying out the body plan of a developing human embryo, okay? So let's take this sort of compare and contrast example with the fly a little bit further. The human genome consists of approximately 3.2 billion base pairs of DNA. Remember, a single base pair just simply means that if you were to look at DNA as a double helix, like a ladder that's twisted around, the rungs of the ladder in the middle, you now all are familiar with, are pairs of A's across from T's, C's across from G's, right? So each one of those rungs on the ladder is a pair of nitrogenous bases, so base pair, right? So our genome across the 22 autosomes and set of sex chromosomes, you would count out 3.2 billion of these base pairs. And as I said, we have somewhere between 20 to 25,000 genes. Why am I ambiguous about 20 to 25? Why can't I just say it's either 20 or 25,000, right? It's because, remember, each one of these molecules is nothing more than just a big strand of A's, T's, C's, and G's, right? So it's very challenging to try to figure out, if you're looking at a length of DNA, whether you're really looking at something that represents a gene, or is it just a length of, DNA, right? That doesn't actually have any significance. So over time, every year, we get a better and better sort of estimate on how many genes we actually have, right? And the definition of gene can get a little bit ambiguous as well. So let's just say, for argument's sake, we have 20,000 genes contained in the human genome, right? And that's all of the separate instructions that your cells of your body need in order to be able to become one of 220 or so different cells that you have. Let's look at and compare that to our friend, the fruit fly, the Drosophila. So the Drosophila genome is quite a bit smaller. Instead of 3.2 billions of base pairs, Drosophila's genome is like 122 million base pairs, right? But what's interesting is the number of genes it has isn't proportionally that much smaller. Drosophila has about 15,500 genes in its genome. And together, these genes encode all of the information that it needs to have different cell types that give rise to the digestive tract, the body organization, nervous system, and other systems like circulation, skeletal system, muscle system, etc., just like in people, right? So here's the sort of lowly Drosophila genome. Drosophila genome is organized kind of weird. There's actually 4 pairs of chromosomes. The 2 pairs of big ones are actually broken into a chromosome 2 that has 2 parts and a chromosome 3 that has 3 parts. All right. So consider this. We as people have a 30-fold larger physical genome in terms of number of base pairs compared to a fly, but we only have like 30% more genes than a fly does. Right? So all of this huge extra physical DNA in our genomes, right? And yet, the number of actual significant regions of DNA that are genes isn't that much bigger. So what does this tell you about gene regulation, right? And the actual number of genes versus the size of a genome? As it turns out, we now know that much of our genome is actually regulatory, not coding. What do I mean by that, right? If you were to actually look at the physical locations of the 20,000 genes that make up the human genome, right, that would only take up about 3% of the actual physical amount of DNA that's in there. That means the other 97% or close to 98% is almost like what? Junk or filler material, right? Not important stuff. Well, it's true, right? A few decades ago, we thought that that's what it was. We thought that like 97% of the human genome is just junk. It's just filler DNA, and the parts that actually code for meaningful genetic data was only like 3%. But now we know, right? We were very, very naive about that, right? Because that 97% of the genome is regulatory. Now, what do I mean by that? Regulatory implies what? It has some sort of control function. And that's indeed what we're talking about. Remember when I showed you out of my wallet a big long receipt from the Long's drugstore cash register that has all these coupons on it, right? And I said, let's pretend that that's a big long strand of DNA like a chromosome. And I said that, you and I, because we can read, right, can decipher or interpret where one coupon begins, where one coupon ends, and the information in that coupon. But that's based upon the fact that we have a very sophisticated system of interpreting a lot of different types of data at once. In the case of the cash register receipt, it's what made up of things like different fonts, different bold versus normal shading, 26 letters to choose from to spell things out, 10 digits, 0 from 9, right? And punctuation marks. Right? So because we have a complex amount of different types of data, right, we can interpret it. But DNA is just made of A, T, C's, and G's. So that means if you don't have a lot of variety in the different types of units of data that you have, right, how can you get super complicated instructions from that? You would do it because the cell would rely upon the rest of the genome, these other 97% of stretches of DNA, to provide essentially the control instructions on how to actually interpret and make use of the 3% of the genome that are the genes. All right. So in other words, the simpler the starting data is in terms of just being A, T, C's, and G's, right, the more complicated the instructions are that have to go along with it in order to interpret it, right? And those instructions turn out to be also encoded in the A, T, C's, and G's of the bulk of our genome. So hopefully you find that you follow that and you're not like, well, I totally don't get what he's talking about. Let's start off, okay, by learning how some simpler examples of genes are regulated in living things on Earth. So let's start off with prokaryotes like E. coli. So this is E. coli. It's a prokaryotic organism. It's a bacteria. Researchers, of course, have used E. coli as a model organism for understanding genetics and how living things utilize genes. So the E. coli has a genome size that's comparatively tiny to ours. Instead of like several billion, right? It's like 5 million base pairs. That's it. Again, just one long string of A, T, C's, and G's on one circular chromosome, right? Because that's what the genome of a bacteria is. Right? And we now know it contains, in that long sequence of bases, 5,400 different segments that can be considered separate instructions or 5,400 separate genes. So this E. coli genome shown over here is drawn as a circle. So this map is a way of conveniently showing, right, the 5.4 million base pairs drawn in a circle, and all of these different sort of data readings of peaks and valleys, stuff like that, are different types of things that researchers have identified along the length of that 5 million base pairs. Right, so as you sort of move out in this sort of concentric type of a map, it's just different layers of data, right? You do not need to fully understand what these different data are, but I think what I want you to appreciate is that this is a convenient way to have lots of data sort of all overlapping on each other to form this complete map of the E. coli genome. The outermost ring has some examples of very important genes that are represented now in these colored blocks, right? So some of these might be responsible for giving the E. coli different types of pathogenic properties. So this E. coli strain is the one that's often indicated in food poisoning, for example, right? So you better imagine that researchers want to understand what types of genes in the genome of this food poisoning E. coli, right, are actually responsible for making things like the toxic proteins or things like that that make us sick. Right? Okay. So using bacteria as a good example, right, is a nice place to start because its genome is so much smaller than ours. Right? Instead of 20,000 genes, it has 5,400 genes. Right? But many of the mechanisms by which the E. coli uses for gene regulation, in other words, deciding which genes at what time it should actually express, right, through transcription and translation into finished proteins, can serve as a really good model for understanding more complicated processes in our body. So, all living cells have to undergo metabolic processes, right? Metabolism. Metabolism is essentially the combination of taking complex molecules from the outside and breaking them down into smaller molecules that can be used as raw material to make the macromolecules that a cell needs to survive. Right? Metabolism is not only the breaking down of big, complicated molecules into smaller pieces, but it would also involve the assembly of smaller pieces into new macromolecules, the ones that the cell in particular needs at that time, right? So at that moment, it's worth it for the cell to break down big molecules to get raw materials, to reassemble them, to make a different type of, let's say, macromolecule, one that the cell needs in order to deal with things like its environment or what it's being exposed to or what types of processes the cell is trying to accomplish. So let's start by using metabolism as an example to understand basic gene regulation. Right? So catabolism is the breaking down of molecules for raw materials. So the way that something like an E. coli would break down sugars, right, that it has access to from the outside in order to get things like glucose from the sugars for energy is a good example. So let's say you start off by looking at the lactose pathway. So you remember lactose is a disaccharide. This is essentially the milk sugar, right? So it consists of a molecule glucose and a molecule of galactose together to form a disaccharide. Right? Let's look at how this pathway involves several proteins in the E. coli working together in turn to get the glucose molecule off of the lactose molecule. So there are three critical proteins involved in this. They're labeled 1, 2, 3 here. All right, let's start off with the first one. There is a protein known as galactoside permease. Okay. So again, all of these proteins that function as little machines, they end with ACE because technically they are enzymes. We will learn more about enzymes later, but for now, right, molecular machines that are proteins that function as enzymes, they end with ACE in their name. So here we have a galactoside permease. As the name implies, its job is to take lactose from outside of the cell and allow it to permeate or come in through the plasma membrane. So here is the ribbon diagram of that enzyme. I don't know for sure if this is a lactose molecule represented in there, but for our sake, we can imagine that it is, right? So this protein brings the lactose into the cell. And here's the lactose, right? It's made up of 1 glucose, 1 galactose. The next enzyme that's involved, number 2, is this transacetylase. And what does it do? Like the name suggests, right? It's going to somehow move an acetyl group onto something else, right? That's the trans part. So here's an acetyl group. It's going to add it to this lactose. Why? Because remember, if you add a small group to a small molecule, you end up changing its shape, right? So from that moment on, it's technically not lactose, right? Because it now has these extra atoms on it, right? And that gives it the right shape for the third enzyme, this beta-galactosidase, to recognize that shape, be able to bind to that acetylated lactose shape, right? And break the glycosidic bond between the two sugar monomers, thereby releasing the glucose. Right? And now the glucose can be used by the bacteria for energy, for example. Right? So you see why we call this a pathway, right? Where you have different proteins that all have to act in turn in order to accomplish sort of the next step of that pathway, ultimately leading to being able to get a glucose off of the lactose. I want you to consider this. In order for this to be a very efficient pathway, that means the production or the presence of these 3 different enzymes that all take turns working on it right? That has to be expressed or regulated in a coordinated fashion. It would not make sense to make a ton of enzyme number 1 if you're not making any of, let's say, enzyme 2 or enzyme 3, right? And likewise, what good is it if you made a ton of enzyme 3 if you're not making enough of enzyme 2 in order to make this pathway connect? So one way that genes that code for these different proteins can be coordinated is just to have their on-off status, whether they're being expressed or not, sort of synchronized. And a convenient way of synchronizing that is just having the genes physically close to each other under the control of a same identical promoter region. Right. So this type of arrangement of having genes that are involved in ultimately making proteins that are going to work together in a coordinated fashion in some critical cell pathway, right? Having these genes located together in bacteria in the genome is what we call an operon, right? So this set of genes functions together almost like one. So genes with related function that are grouped together under the same control. So this blue cartoon in the middle here, this represents, right, a zoomed in part of the E. coli genome. So now we've zoomed way in there and we see this particular stretch of the genome contains these 3 genes, LAC-Z, LAC-Y, and LAC-A, right, next to each other. And they're all under control by the same promoter region. Remember, the promoter is where the RNA polymerase is going to dock here and end up transcribing, right, this whole operon to give you one mRNA, right, that then can be divided into different parts, each of which is responsible, once it's translated by a ribosome, to make the 3 different enzyme proteins, right, that all work together to make the catabolism of lactose functional. So think about this for a moment, though. If you have E. coli floating in a puddle, right, by the side of the road, is there going to be a lot of lactose sugar for the E. coli to work on in there? No, right? There might be very little or no lactose floating around in that puddle by the side of the road. So as a result, wouldn't it be an awful waste of time and resources if the E. coli kept making these different lactose pathway enzymes instead of maybe using those raw materials to like make other proteins, right, that would actually help it survive in the puddle? Right? So this has to do then with gene regulation. So notice the promoter region has this little tan colored region next to it labeled O. We call it the operator. Okay? And that operator is going to influence whether this promoter region works. It's going to function through the influence of another gene that's upstream of this to the left, known as the LAC-I gene. So let's look at how this works. Okay. Oh, so right. The bottom line is the arrangement of genes this way in an operon is going to give this coordinated on-off expression of the different pathway components. All right. So the way it works is this. Normally, in the absence of lactose, so there's no lactose surrounding the E. coli, so there's no reason for the E. coli bacteria to even want to bother making these lactose processing enzymes, right? Because that'd just be such a waste of time, and more importantly, a waste of raw materials, right? Like amino acids, right? They could be assembled to make other proteins instead. So normally, in the absence of lactose, there is a protein here labeled R that's a repressor that binds to the operator sequence and gets in the way of RNA polymerase being recruited to the promoter. Where does this repressor protein come from? Well, it's a protein, so it must have been translated from some mRNA. And sure enough, that mRNA is what's transcribed off of that LAC-I gene. Okay? So this LAC-I gene is always expressing and making this repressor. And this repressor is the right shape as it's floating around in the cell. Some of these repressor molecules will bind to the operator and keep the LAC operator turned off, or the LAC operon turned off. right? So you can see in this ribbon diagram down here, here is in yellow is the little section right here. That would be the operator sequence. Okay. This big blue and red protein is the repressor protein. You can see it binds to the operator right here, and this gets in the way of RNA Pol II. Okay. So this repressor protein has a binding site in it for lactose. You're like, why would that be important? Well, let's look what happens, right? So in the absence of lactose, the repressor protein binds to the operator and keeps the production of the LAC-Z, LAC-Y, and LAC-A genes turned off. But if suddenly you get a lot of lactose surrounding the E. coli, right? So what about this E. coli suddenly ended up in a carton of milk, right? Whoa, that's a lot of lactose. That's a lot of good sugar for this E. coli to make use of. If only it had the different enzymes available inside of it to make use of it. Right? So the lactose molecule itself, the green ball here, binds to the repressor protein because, again, I said the repressor protein has a lactose binding site, and that causes the repressor protein to change its shape. And when it changes its shape, it no longer binds to the operon DNA, right? It essentially falls off, and now RNA polymerase can be recruited here and start transcribing the operon, right? So it makes the mRNA in red here that then can be cut into the different parts that end up being translated into the three important proteins that now can start doing things like taking the lactose from the milk into the E. coli, processing it, like putting the acetyl groups on it that make it the right shape for the third enzyme to come and break the lactose. And so now the E. coli is getting glucose from it. All right. So, I know that sounds very complicated, but at the same time, hopefully some of you, when you look at this, you're like, wow, that's actually very clever, right? So the presence of lactose, right? In other words, something for the E. coli to actually work on, is the stimulating signal that then allows the E. coli to make the enzymes in it that will enable it to work on the lactose. Right? Okay, here's another example of a regulatory protein. This is another example of a protein that helps with this lac operon control. This is cyclic adenosine monophosphate, or CAMP. If you look at the structure here, you're like, well, that looks kind of familiar, but it's kind of weird at the same time. Why does it look familiar? Well, you realize this is just a modified nucleotide, right? Here's your nitrogenous base up here. This is the adenine. Here you have a ribose sugar ring, right? And then here's a phosphate group attached to it. But here's where things get weird, right? Because instead of the phosphate group just being attached through the C5 position, and the phosphate group would then normally be available to link up with another nucleotide if you were putting together an RNA strand. Look what's happened here, right? This oxygen of the phosphate group has now bound to the C1, 2, 3 position. So you form a little cyclical ring over here. So that's why it's called cyclic AMP. As a result, this particular nucleotide won't work to be a building block of a long chain of RNA, right? But Mother Nature, as I've said before, right, in evolutionary history, when life stumbles upon different molecules that are useful for something, like nucleotides to build long nucleic acids, right, evolution, Mother Nature finds other uses for these molecules as well. So in this case, converting this into this cyclic form, right, makes a very important molecule that has to do with sugar metabolism, not just in bacteria, but in our livers. So here you can see, here is cyclic AMP, right, in yellow. So that's this molecule up here. There's a protein known as catabolite activator protein, or CAP. And CAP is the right shape to bind cyclic AMP, and that changes the shape of the CAP protein just enough that two CAP proteins, these blue things here, each bound to a cyclic AMP, are now the right shape to come together and bind to DNA. Right? So where is it binding in this lac operon in the bacteria? Here you can see, right, the cyclic AMP activated cap protein binds to a site, the cap site, this light blue stretch of DNA, right? That is essentially just upstream of the promoter region for the lac operon. And when this cap activated protein binds there, it helps shape the DNA in such a way to make it even more attractive for RNA polymerase to come and dock here and start transcribing. So in other words, the presence of cyclic AMP encourages this protein that binds to the cyclic AMP to then help with the recruitment of RNA polymerase to drive expression of the lac genes, right? So if you connect the dots, that means the presence of a lot of cyclic AMP encourages ultimately the E. coli to be able to process things like lactose, right? So glucose, though, remember the thing that ultimately the E. coli wants to get off of the lactose, is sort of in a feedback loop that discourages the production of cyclic AMP. Why does that make sense? It's because if there's a ton of glucose already available, again, why would the E. coli want to spend time and resources worrying about breaking down lactose to get glucose out of it? Right? So if you already have a ton of glucose, a ton of glucose actually inhibits the formation of cyclic AMP. And so that's another way that glucose can actually keep the lactose pathway sort of shut down. And as I said, cyclic AMP is not only important in this E. coli example, but our liver uses cyclic AMP to help break glycogen, right, down into individual glucose molecules to be released into our blood. Right. That's kind of amazing, right? When you think about it, something that's in bacteria that works this way, this molecule, cyclic AMP, right, to this day is also used inside our bodies to handle sugar metabolism. Right. So here's another illustration of the lac operon. So this would be the region close to the promoter of the lac operon where that big repressor protein binds to, right? And you can see the repressor protein interferes with RNA polymerase being able to see this because the repressor protein actually causes the DNA to kind of get looped up, right? So that RNA polymerase can't bind here. But you can see the cap protein is already kind of present in there, right? So once the repressor protein is kicked off, because now you have a lot of lactose coming in, right? And the lactose binds to the repressor protein comes off, the DNA kind of unravels, right? And then this cap protein is already there. So it helps encourage RNA Pol2 to come to the promoter and you start getting expression. Right. The last example for today, right, is sort of the opposite. So breaking down lactose to get glucose out of it was an example of catabolism, right? The breaking down of a larger molecule to get something smaller. But as I said, a cell is always putting together small raw materials to make things that are more complicated that it needs, right? And that process of building up is called anabolism. So anabolism is the creation of new molecules from raw materials. So depending upon how important these products are that these anabolic pathways are making, they may be things that just need to be on all the time. For example, here's another operon in E. coli. This is a set of genes, right, that make enzymes that ultimately allow the E. coli to make the amino acid tryptophan. Remember, tryptophan is one of the 20 amino acids that the E. coli, just like us, need to have available to be able to build things like proteins. So you can imagine then there's never a time when the E. coli doesn't need tryptophan. So the enzymes that are involved in that pathway, that anabolic pathway to make tryptophan, are just always on unless you really don't need, right, to have so much tryptophan being made. So in principle, it's the same as the lac operon, but it works oppositely. So in this case, you would have a repressor protein that is made by some tryptophan operon repressor gene. But in this case, this repressor protein is the wrong shape to bind and block the operon, right, and block RNA Pol2. So this repressor protein is just floating around. And meanwhile, right, this big purple RNA Pol II keeps visiting the promoter and keeps transcribing and making ultimately all of these RNAs of these different TRP pathway genes. So the cell is always making all the enzymes. So it's always making tryptophan, right? But if you get a ton of tryptophan, like the cell has way more tryptophan than it needs, again, you might as well turn this off, right? So that you can use your resources to make something else. So here's the tryptophan. And in this case, when it binds to the repressor protein, now it makes the repressor protein the right shape to bind to the operon and block the RNA Pol II. And so here's the TRPR protein. So over here, this is the TRPR protein. And once tryptophan comes in, right, it changes its shape. And now that can bind to the operon stretch of DNA to shut down this operator. Right? So the take-home messages from today are that catabolic pathways get stimulated by the presence of the starting material that you need to work on. So for example, the lactose metabolism pathway, right, is stimulated by the presence of lactose, whereas anabolic pathways like the tryptophan one get repressed if you have too much end product as a negative feedback loop. Okay. All right. So those can be kind of confusing, but again, talk your way through it, right? Try to understand the principles of what I'm explaining here, right? How these genes can be controlled by other proteins binding or not binding to these regulatory sites, like the promoters. Right? Okay, so let's stop there and I'll go to the chat if there are any questions. All right. Okay, I don't see any questions in the chat today. So if you have anything that you think of after you've had a chance to look this over, ask your TI about it. He or she, they know this stuff really well. And if there's something that they don't know the answer to, feel free to reach out to me and I'll be happy to explain it to you either over email or during office hours. Okay. All right. Have a good day.